Welcome to part 2 of chapter 4. So I'm going to start by going over neurons, and then we'll wrap up the chapter by getting a little bit more specific about the brain. So to start, the brain has two different types of cells. So the first, neurons, are the ones that we're going to mostly focus on because they are considered the communication specialists of the body. They're essentially the building blocks of the nervous system. But we also need to at least acknowledge the role of glial cells because they used to be largely ignored. We used to think that they were just glue that kind of held the nervous system together, and in some way that's not wrong because they do hold neurons in place, but they have a more sophisticated role than that. They also nurture and insulate them. So we do need to acknowledge the role of glial cells in the nervous system because they do serve an important function. So let's get a little more specific about the structure of the neuron. So the first thing is that a neuron has dendrites, which are these kind of branchy, tree-like looking things down here. And so those are going to be the antennas that receive messages from other nerve cells. Once the dendrites receive the message, it's then going to send the message down to the cell body, also called the soma. So the cell body is going to contain the cell nucleus, have DNA in it. It's also going to kind of regulate and control the cell's growth and reproduction. From there, the message continues down onto the axon, and that's kind of the end point of the message because it then is going to be sent away from the cell body, from that particular neuron to other neurons, muscles, and glands. And so we also need to recognize that the way that the chemical message is kind of sent, or the electrical impulses we'll talk about in a second, is it's kind of transmitted down the neuron is going to depend on the exact type of neuron and whether it has this kind of like fatty sheath on it. And so your book does a nice job of kind of highlighting the different ways that message can be sent down the neuron. And so, as again you learned in your book, the neurons don't touch each other, but they are the communication specialists. And so there must be a way that neuron A can communicate with neuron B. And the way that it does that is using chemical messages. And so here you can kind of see, this was really the best depiction I could find. Many of them were much more complicated, but this is essentially, let's say neuron A, we could call it the presynaptic neuron, meaning it's the neuron kind of sending a message to the other neuron, which is neuron B, the postsynaptic neuron. So this postsynaptic neuron is the one that's going to kind of receive the message from this presynaptic neuron. This postsynaptic neuron is at rest, meaning it has not been stimulated and it overall has a negative charge. So this presynaptic neuron is going to release a neurotransmitter from the axon terminal down here into the synapse, and so that chemical message is going to go across the synapse to the postsynaptic neuron. That postsynaptic neuron is going to receive the neurotransmitter via the dendrites. Okay, so essentially what happens is the neurotransmitter is going to bind onto the dendrites of the receiving neuron. And so what ends up happening is this is going to open up channels that allow positively charged sodium ions into that postsynaptic neuron. Okay, and that essentially excites the neuron. And so we talk about that in terms of depolarization. If that cell becomes depolarized enough, then we reach what's called an action potential. Essentially, that just means the neuron's going to fire. Okay? And so that action potential is going to travel its way down the axon, okay, and eventually reach the end of that neuron. Once it reaches the end of the axon, where the neurotransmitters are stored down here in synaptic vesicles, it's going to then release the neurotransmitter into the synapse to then bind with and communicate with another neuron. Okay, so this is, again, very simplistic because we only have two neurons here. In actuality, as you learned in your textbook, there are many, many more than that in the human body. And so this is a very simplified version because this is going to happen, relatively quick. Multiple neurons firing in sequence to send important chemical messages. So that's essentially, how the message is going to be sent down from the dendrite through the cell body, down the axon, and again, talking about positivity and negativity in terms of the action potential. So I just wanted to kind of zoom in here. We're focusing in on the axon terminal of the presynaptic neuron at this point. So let's go back. So we're going to kind of focus in here, and we're going to focus on the ends of the axons of this presynaptic neuron that's going to send the message. So again, what happens is that action potential is going to travel down the neuron and reach this end here where the neurotransmitters are going to be released from the synaptic vesicles. They're going to enter the synapse and move over and bind with the receptor sites of the dendrites of the receiving or postsynaptic neuron. But I also want to point out that what can happen then after that is the kind of leftover neurotransmitters will then go through what's called a reuptake process, where they're essentially reabsorbed back into that presynaptic neuron. And so we talk about reuptake a lot in terms of medications. Think about SSRIs, which you might have heard of, are often used to treat depression. It's a selective serotonin reuptake inhibitor. Okay, the most important part there for right now is reuptake inhibitor, meaning the medication essentially acts by blocking the reuptake process so that the neurotransmitter hangs around the synapse longer, allowing more opportunity for it to bind with the receiving or postsynaptic neuron. This is a super complicated process. This is a rather complex concept. And so, if this is kind of hard to get the first time, I totally understand. Definitely sit down and look at your book again. We're not going to get too complicated in this class. Just kind of acknowledge and understand that this is a rather sophisticated area of psychology, but it's important to understand because, neurons and neurotransmitters are kind of so fundamental to the nervous system communicating. So speaking of neurotransmitters, I just want to kind of mention a few of them because, to really kind of appreciate how we treat psychological disorders, for example, you have to have an understanding of neurotransmitters. And so later in the semester, we will talk about treating psychological disorders. And so here I have just kind of the one shot, just a few words to describe the main neurotransmitters we're going to be focusing on. So serotonin, it's involved in sleep, it's involved in appetite, sensory perception, pain suppression, mood. And so if you kind of remember what I just said about what SSRI stands for, it's serotonin selective reuptake inhibitor, meaning that one of the ways that we treat depression is that we alter the activity of serotonin in the brain, essentially. Dopamine, on the other hand, is involved in voluntary movement, attention, learning, memory, emotion, pleasure, basically how people respond to novelty as well. And so dopamine has been implicated with Alzheimer's. It's been connected to schizophrenia as well. Acetylcholine, that's a lot of things related to like arousal, vigilance, memory, emotion. Norepinephrine is related to learning and memory, dreaming, wakefulness, emotion, can also impact heart rate. Oh, and also can impact digestive functioning, particularly during stress. GABA is an inhibitory transmitter, meaning like suppressor, and then glutamate is a major excitatory transmitter, meaning that, it's going to basically activate brain activity, and it's actually released by about 90% of the brain's neurons. So this is just like a snapshot of what neurotransmitters can do, and the field is often in conflict about how are these neurotransmitters truly linked to things like depression. So I know there's a little bit of a debate in the clinical psychology field about what really is happening with serotonin. Is it truly that the levels of serotonin are too low? Is it more about like activity or the receptor activity related to serotonin? So this is very complex. And so we're, again, kind of making it overly simplified. And so I want you to, again, kind of acknowledge that, but I don't want the class to get too complex. So we have a lot of really cool kind of evolving techniques for mapping the brain. So we know a lot about different structures in the brain, where things are housed. We know a lot about the function of different areas of the brain, like what becomes active when we're thinking, what becomes active when we're, emotional. But I also want to kind of, give appreciation for that. This is very complex, and the way that we're interpreting these results often make it overly simplified. So I know I said I was going to stick really just to whatever was in the chapter for this PowerPoint, but I did want to include this because I think that it does kind of add something extra, and I don't think your textbook talks about it. So there was a group of graduate students a few years ago that were just bored in their research lab one night, and they had been doing fMRI research, which you can see, here's an example of what a scan can look like. And so you're essentially looking for brain activity. So you see those little red dots, it means that that area of the brain was active during whatever task the participant was doing. So they were bored in the lab, and they decided to go to the grocery store and get a dead salmon from the meat department. And so they took this dead salmon and they put it in the fMRI machine. And I'm not exactly sure what they were trying to do. I think it was more of just kind of, they were bored and joking around. But they were shocked when they saw the results, because this is the scan. As you can see, there is activity in multiple areas of the dead salmon's brain. So this is pretty problematic, because a dead salmon that had been in a grocery freezer, for who knows how long, should not have brain activity, and yet it did. And so what we learned from this is that oftentimes we get kind of fluke results. So think back to when we talked about the method of psychology and the science behind psychology. We sometimes get just random findings. And so one of the things that we're doing with this type of research is anytime you do a brain scan, it's essentially comparing every little pixel of the brain to all of the other pixels of the brain. So it's calculating millions and millions of tests, statistical tests. And so as we talked about in the earlier chapter about methodology, we make mistakes when you're doing that many tests. By just random chance, you will find a result just because of probability. And so that's what ends up happening here. And so this isn't to say that brain research is 100% flawed. It's just to offer caution that don't fall for the dead salmon mistake and assume that these results are 100% trustworthy. We always look for replication. Remember, replication from that chapter simply means we do the study several different times, several different ways before we feel like we can truly trust the results. So if we can replicate it, then that means that we can have more confidence. So I would bet some money if you put a dead salmon into an fMRI machine, you're not going to find activation in the same areas. You might find activation, again, just because of random chance, but they wouldn't be in the same parts of the brain, which means we can probably rule out that any of these salmon are actually thinking. So this kind of relates to the next part about how complicated the brain is, and it is very tempting to oversimplify things. So in your textbook, they talk about localization of function, which essentially acknowledges and talks about that different parts of the brain are going to perform different but overlapping tasks. So that means that through this type of research where we, measure neural activity with electrodes, or we use magnetic fields to look for brain activity, we can kind of isolate and talk about particular parts of the brain being primarily responsible for particular tasks. That is not to say, though, that those areas of the brain work in isolation. We have to be very mindful that all of these parts of the brain come together to kind of orchestrate these more complex tasks. And so we cannot oversimplify this. But research has been pretty effective in determining areas of the brain that are primarily responsible for particular functions. And so several sections of your book go through that in great detail. This is the section of the chapter I think gets a little overwhelming. So I'm not going to do a ton to kind of repeat what your textbook did. So please make sure you read your textbook and that you're familiar with, for example, what the thalamus does, what the cerebellum does, the different hemispheres, the lobes. Please make sure you're familiar with that. But to me, that's simply you sitting down and kind of learning it and memorizing it on your own. What I would rather do is kind of give you an opportunity to test your knowledge in a way that maybe moves it past what the textbook is teaching you. And so what I've done here is I've written several questions. So once you kind of are ready, what I would do is just pause this, read through the questions, write down your answers, and then I'm going to start the next video, so part 3, with the answers. So don't listen to the answers until you kind of think through it, because I do think it will give you the opportunity to kind of think through and apply, because most of these are application questions that I really will, I will include some of these or versions of these or similar questions on the timed quiz 4. So please make sure you're ready to answer these types of questions. So what I'll go ahead and do is kind of stop it here, and then I will go through the answers in the last video, which is part 3.